Good afternoon, everyone. I welcome you all for my second session of bone tissue engineering. In our first session, we discussed about the function of bone, the brief introduction about what is bone and its function, and we studied its anatomy and physiology in detail, and also the formation of bone as well. So today, in this session, we'll be discussing about bone tissue engineering strategies and what are the key components present in tissue engineering application. So in the last 3 decades, bone tissue engineering has been a very new concept and a useful concept for orthopedic surgeons as well as the biomedical community in order to overcome the limitations for the enhancement and regeneration of bone defects. So to start with, we should know what is tissue engineering. I think by this time you all would know what is the definition of tissue engineering, which is nothing but where it encompasses the knowledge of life sciences and engineering together for the development of a biomaterial in order to replace, restore or regenerate a diseased tissue or organ. Or in general, it has been defined as the application of scientific principles to the design, construction, modification and growth of living tissues. There are 3 major elements present in tissue engineering which comprises the tissue engineering triad. The first element is biological scaffold, which is biomaterial, and the second one is cells, and the third one is growth factors. So scaffold, cells and growth factors all together makes a tissue engineering triad. So this is just a brief introduction about tissue engineering and tissue engineering triad. So what is the need for bone tissue engineering? I think we have discussed that in our first session as well. So to overcome the limitations which we are facing in current clinical operations, like in grafting procedures like autograft, where we graft tissues from the same individual, but due to limited availability, donor site morbidity, and the second option is allograft, where it has the chances of immune rejection and pathogen transfer. So in order to overcome all these limitations, we need a substitute, bone graft substitute, to enhance the bone regeneration or to repair the bone defect. So the field of tissue engineering, bone tissue engineering, focuses on the alternative option that will completely eliminate the above said limitations which are facing in the current bone grafting procedures. As I said, like over 2 million procedures, surgical procedures is performed in every year, bone grafting procedure, where bone is considered to be the second mostly transplanted tissue after blood. So this diagram explains the classic bone tissue engineering paradigm, which highlights the several key players. The key players are the 3 major components of tissue engineering triad. The cells, the first one is biocompatible scaffold that closely mimics, the scaffolds are nothing but the 3D structure which mimics the extracellular matrix. Scaffolds and the osteogenic cells to lay down the bone tissue matrix. When I was explaining about the cells, types of cells present in the bone, we have 4 different types of cells, where osteoblast cells, osteocytes, osteogenic cells and osteoclast cells. So osteoblast is responsible for the formation of bone, osteoclast is responsible for bone resorption, and osteogenic cells is the only bone cells that can differentiate into osteoblast cells. So we need osteoblast cells, osteogenic cells to lay down the bone tissue matrix, the second essential component. And the third one is the growth factors, which is the morphogenic signals that can help to direct the cells to the phenotypically desirable type, and also for the growth, enhancement of the growth. That is, the growth factors is the third major component of bone tissue engineering. So scaffolds, cells and biological factors, all 3 together to comprises the tissue engineering triad, bone tissue engineering triad. So in this session we will be dealing about in detail about the each component, key component of bone tissue engineering. So bone tissue engineering involves the uses of porous 3D scaffolds. The 3D scaffold, as I said, it mimics the extracellular matrix, or in general it acts as the template. It acts as the structural template for the cell to enter, adhere, proliferate and differentiate, and thereby it enhances the formation of new tissue and it has to degrade. So, it should be degradable, and it is a temporary implant. It cannot be assigned to a permanent implant, and so it involves the uses of porous 3D scaffolds that along with cells and bioactive factors that can provide support for cells to spread, migrate, differentiate for new tissue formation. Sorry. Three components are biomaterials, cells and growth factors. So, we will be seeing the strategies for biomaterials, cell based approaches and growth factor based approaches in the later presentation. So, to start with biomaterials based approach, we should know what is a biomaterial. What is a biomaterial? Biomaterial is a natural or a synthetic substance that has been engineered to construct to perform in a to perform a biological function with a medical purpose. Okay, it can be either medical purpose can be either therapeutic or a diagnostic one. Evolution of biomaterials. In 1960s or 1970s, first generation biomaterials has been developed. So, it was developed in an idea just to mimic the tissue which got damaged due to physical reasons, or where it has to mimic the physically damaged tissue because of fracture or disease or any other traumas. So, they are just a bio-inert materials and they do not interact with the biology of the host organism. So, first generation materials are called as bio-inert materials. For example, stainless steel and its alloys comes under bio-inert materials, and then they switch the gears from the passive materials to the active materials. The second generation materials are called bioactive materials. Bioactive materials, where it can interact with the host biology, host organism biology, where there will be an interaction between the grafts as well as the cellular level interactions between the host organisms. And in 2000s, third generation of biomaterials has been developed, where it is the bioresorbable materials, where it helps in the formation of new tissue. It combines the properties of bioactive as well as the bioresorbable, where it interacts with the cellular level in the host organism, as well as it gives a specific response, and also it develops the new tissue formation. It helps in the development of new tissue formation. And in 2020s, where they develop biomimetic materials, where they develop a material which mimic the nature of the natural material. So, in this biomaterials based approach, we will be focused mainly on third generation materials. For example, polymer and its composites. So, again they are classified into osteoinductive materials, hybrid materials, advanced hydrogels, immunomodulatory materials. The first category is osteoinductive material. The word osteoinduction, which means it instructs the surrounding cells for the formation of bone, surrounding stem cells for the formation of bone. So, those materials should have the ability, if I say this is an osteoinductive material, that means that material has the ability to induce the bone formation by instructing the surrounding cells in vivo environment to form bone. For example, hydroxyapatite and calcium phosphate or ceramic based materials are prone to have osteoinductive property. The second generation class of biomaterials is hybrid materials. They are nothing but the combination of two or more biomaterials with enhanced functionalities either in the form of copolymers or polymer-polymer blend or polymer ceramic composites. Copolymers are nothing but it is a substance where it developed from two or more monomeric species. For example, PLGA, where it developed from the monomeric units of polylactide and polyglycolide. We can tune the properties of both the monomers, where PLA has the glass transition temperature above room temperature, and it has very long degradation rate, whereas PGA, polyglycolide, has glass transition temperature below room temperature and it has shorter degradation rate. So, we can combine and tune the properties of PLA and PGA, which are FDA approved polymers, and we can develop a polymer which is copolymer hybrid material PLGA with a tailored property in the end, and polymer blends. So, again the same example, I will tell you about PLGA. The degradation product of PLGA will be acidic in nature. So, prolonged exposure of tissue to acidic product will lead to tissue necrosis, and eventually the implant will lead to failure of implants. So, what researchers will do is they combine with the polymer, like for example phosphozines. Phosphozines, where the degradation product of phosphozines are in neutral pH. So, they combine PLGA as well as phosphozines to develop a biomaterial to give a non-toxic degradation products. The final one is polymer ceramic composites, which are very useful in bone tissue engineering, and this we can call it as biomimetic scaffolds. It has all the desirable properties for bone tissue engineering. Then third class of biomaterial is advanced hydrogels. Hydrogels, because of its physical properties and its structure, it has been widely used in tissue engineering application, and the recent research have showed that self-assembling peptides have gained recent enormous attention for forming scaffolds, as they are completely biological, biocompatible and biodegradable. For example, RADI16. It is a self-assembling peptide where it aims to mimic natural extracellular matrix, and it can be readily synthesized, and it is injected in the form of nanofibers. It is injected in the form of nanofibers. Once it enters the physiological fluids, and it becomes this gel, and it serves as a template for the starting material. Template as a starting material. The final one is immunomodulatory biomaterial. So, in order to suppress the immune reaction, we can develop a biomaterial that can modulate or manipulate the immune system in a favorable manner for the enhancement of bone regeneration. So, this is all about the biomaterials based approach, where I will focus mainly on scaffolds. The scaffolds are the masterpiece of bone tissue engineering. A bone scaffold is the 3D matrix that allows and stimulates the attachment, proliferation of osteoinducible cells on its surfaces, and the first one was developed by Green in early 1970, where they seeded cartilage cells into the scaffold. That is how it has developed seeding cells on scaffolds. And the image which shows there is a long bone where there is a defect, and we have to place a scaffold in that defect and check for the enhancement or regeneration of the long bone. So, scaffold, since the scaffold it is in a masterpiece of the bone tissue engineering, we should be more careful in selecting scaffold and its properties, the materials, and for everything for the preparation of scaffold. So, the first one, the scaffolds can be categorized into 4 classes: polymeric, ceramic, composite or metallic scaffolds. Polymers, again polymeric scaffold can be derived from either natural polymers or synthetic polymers, and composites may be the combination of polymers and ceramics. And the scaffolds requirement of the ideal scaffold, and any ideal scaffold for bone tissue engineering, it should be biocompatible, and it should be biodegradable, and it should not be a permanent implant. It is a temporary implant, and it is non-toxic and highly interconnected porosity. This porosity plays a very important role in bone tissue engineering, because the pore size is very important for the cells to enter and proliferate, differentiate, and also for the diffusion in and out of the nutrients and wastage. The pore diameter or pore size, it should be greater than 100 micrometer. And it should be mechanically strong. Since we are aiming for bone tissue engineering, the scaffold should be ideally strong, and it should mimic the either cortical, if in case for a cortical bone tissue engineering, it should have the mechanical properties similar to cortical bone, whereas the spongy bone, the cancellous bone, it should mimic the properties of the spongy bone. Also, it should enhances vascularization. Vascularization is the formation of new blood vessels. So, any ideal scaffold for bone tissue engineering, it should be biocompatible, it should not be toxic, it should be biodegradable, it should have highly interconnected porosity, it should be mechanically strong, and it should enhances vascularization. The three properties of scaffolds which clearly shows the ideal scaffold properties. The osseointegration, which is nothing but the biocompatibility, it should get integrate well with the ost tissue, and osteoconductivity, where it helps in the formation of new bone on the surface of the grafts, and osteoinductivity, it should direct the cells, surrounding cells for the formation of bone. So, these are the main properties of the ideal scaffold. And micro and macro structures are the nothing but the porosity structures, mechanical properties, it should be mechanically strong, so these are the main properties of a scaffold to be used in bone tissue engineering. So, this picture where it shows the treatment of bone defects with the presence of scaffold. The first, there is a bone defect in the bone gap, defect gap in the long bone, where we place the scaffold in that defective gap, and this scaffold first it should be osseo-integrated. It should be osseo-integrated, then it should be osteoconductive, osteoinductive, and then the healing process. This is nothing but the vascularization, the formation of new vessels. So, from this there will be some few graphical images just for teaching purposes. Should be aware of this graphical images which are taken from the literature for knowing about the information what they have done for scaffold based strategies. So, in these are called as first generation scaffolds, which are nothing but the group in National University of Singapore collaborated with Temasek Polytechnic, where they optimized the parameters for PCL, polycaprolactone, and its composites for the development of scaffold by FDM method. FDM method is fusion deposition modeling, which is nothing but the 3D method, 3D printing method for the development of biomimetic scaffold. They considered this as a first generation scaffolds, where they did perform clinical trials for clinical studies for 5 years. After 5 years, they said that the outcome was positive, and again another research group used this kind of burr hole plugs for the treatment of cranioplasty. Burr hole plugs for the treatment of cranioplasty, the another group has tested with another clinical trial, another set of experiments, where they used this PCL burr hole plugs, and they studied in rats, and after 12 months they got a positive outcome, and the CT images shows the perfect alignment. The CT images shows the perfect alignment of PCL sheets to the 3D orbital cavity. The first image where they use this PCL sheets in the autologous iliac crest, where they removed a skin from there, where they placed this sheet and tested for the regeneration of bone, and they found it successful. And the second where they did for skull defects, the second image where they place these PCL burrow sheets on the skull defects, and then after 12 months of the study they found that they have perfectly aligned and they healed the bone defect. So then they have moved to the second generation of scaffolds, where they wanted to study with PCL and composites. Okay, where they have done they prepared scaffold using PCL, tricalcium phosphate and collagen, and they created a defects around 5 mm diameter in rat parietal bone, and then they tested for healing of the bone defects. And in the C and D you can see the micro CT scanning image, where the skull defect after 14 months there is the complete closure of the bone, there is the closure of the bone with the presence of the scaffold, when it is compared with the blank where there is no scaffold at all. And this was done in rat parietal bone using PCL sheets with and its composites, the scaffolds which these are the biomimetic scaffolds. And this one is the pig spine fusion model, and the below pictures are the rabbit, New Zealand white rabbit skull defects. They have tested for cranioplasty. Again, analysis of tissue engineered construct shows that the formation throughout the entire scaffold and no fibrous tissue formation is seen. And micro CT analysis revealed that the 30% of PCL TCP resorbed and replaced by bone. And this is for the treatment of load bearing osteochondral defects. Again, load bearing, since bone we need to test for load bearing defects, again with the PCL and calcium phosphate, where they have drilled hole in load bearing osteochondral defect. They created load bearing osteochondral defect, where they have seeded mesenchymal stem cells along with the scaffold, PCL CAP scaffold, and then they inserted on the defect. And after 12 months, or I am not sure about the month or the time, after a certain time there is an closure of this bone defect in the presence of scaffold. So this is all about the scaffolds. The scaffolds we have seen about its properties of the scaffold required for the ideal bone graft, and few examples about biomimetic scaffold where they developed for the first time, first generation scaffold and the second generation scaffolds. And now we move on to the strategies for bone regeneration. Cellular based approach. First we seen about scaffold based approach, and now it is the cellular based approach. The cellular based approach, we have seen that there are 4 types of cells. Osteoblast cells, which are responsible for the formation of bone, osteocytes, which are the primary cells for a matured bone, and which are responsible for to maintaining the concentration of matrix, mineral concentration of the matrix, and osteogenic cells, which are responsible for the differentiation and into osteoblast, and osteoclast cells, which are responsible for the bone resorption. And so though the volume of cells present in the bone is less, but its function is crucial. So we need to have bone cells in order to repair bone defects. There are 4 ways. These are the first proposed approach. Firstly proposed approaches, cellular based approaches. Implantation of unfractionated fresh bone marrow is the first approach, and the second one is the purified culture expansion of mesenchymal stem cells, and the third one differentiated osteoblasts and chondrocytes, and the fourth one is the cells that have been modified genetically to have recombinant bone morphogenetic protein. So we will look into first approach. So these approaches mainly based on the primitive mechanism, primarily targets the See, cellular based approach in bone tissue engineering primarily targets the early stages of bone repair when the skeletal progenitors becomes impaired. So, this can be mainly due to either trauma or any disease conditions, or maybe can be due to aging as well. These proposed mechanism by which implanted cells enhance bone regeneration in bone tissue engineering involve. First is early release of key osteogenic and vasculogenic molecules and growth factors, and the second one is it forms the template to recruit the osteogenic cells and vasculogenic cells, and the third one is actively laying down the bone matrix and vascularizing the bone structure construct. These are the steps that follows in the cellular based approaches, and the first approach is the unfractionated fresh bone marrow approach. In the first picture where the patient, the first is they are anesthetized, and the second one is the defect, and where they directly remove the fresh bone marrow and inject the cells into the defective site. So, they remove the bone marrow. So we all know cells present in the bone marrow region. So, they remove the cells from the bone marrow and they inject into the defect sites directly, as well as along with the some matrix. So, they inject bone marrow cells along with the matrix. So, after few days or a few months, we can see from the radiograph images where there is a complete healing of the bone defect. We can see the difference in the picture B and C where there is complete healing of bone defect due to the injection of bone marrow cells, osteogenic cells directly from iliac crest, the posterior wing of iliac crest, and we inject into the defect site. But the disadvantages of this is availability. Again, it is autologous, as well as it increases pain, and it requires 2 surgeries where we need to remove and we need to inject into the defect sites. So, it requires lot of pain and time consuming and everything. And the next approach is differentiated osteoblasts and chondrocytes approach. We all know the bone mechanism, formation of bone mechanism. As I explained in my first session, formation of bone occurs in 2 mechanisms, via intramembranous and endochondral ossification. So, what researchers have thought, like, why don't we inject osteoblast cell, differentiated osteoblast cells or chondrocytes directly into the defect or along with the mesenchymal stem cells. Okay. So, first they have tried, they inject differentiated osteoblast cells, they achieved in that. They achieved increase in enhancement of bone regeneration. Whereas in the second picture, this picture where what they have tried is, they tried to inject chondrocytes. So, because they thought that endochondral ossification, first the formation of cartilaginous template, thereby laying down bone matrix, right. So, what they thought, why don't we inject chondrocytes directly into the defect sites. So, Vacanti and this group, they did this research. What they did is, like, they injected chondrocytes into the bone defect and they compared with the periosteal cells. Where what they found is there was a formation of cartilage layer in both the defects. They compared with periosteal cells and chondrocyte cells, but after cartilage formation, the defect where they inject only chondrocytes, they did not find any formation of vascularization or angiogenesis, nothing. It was like that. In turn, they found that the chondrocytes produces the precursor cells which has the cues which inhibit the vascularization. So, the other defect where they have found the cartilaginous template as well as the formation of bone and everything, it was there. And whereas in the defect where they injected only chondrocytes, they couldn't find the vascularization network or neoangiogenesis, which is the primary formation of blood vessels. So, they found that injecting chondrocytes alone will not support in cell based approach. Then this approach, the mesenchymal stem cells approach, is effective approach where they can this mesenchymal stem cells can undergo a replication without differentiation. It can go and we can passage 30 times of this mesenchymal stem cells, and which increases to up to 1 billion fold of cells without differentiation. It has to differentiate only in the presence of implantation. So, this is one of the research where they recently produced where in the segmental defect they created a bone defect with they inserted collagen matrix. What happened was the endogenous mesenchymal stem cells slightly invaded into the matrix. Then this research group, with the help of ultrasound, they injected BMP, bone morphogenetic protein, in order to start the bone repair process with the microbubbles. So, they suspended in microbubbles and injected into the mesenchymal stem cell populated scaffold, and with the ultrasound what happened, this gene entered into the cells with the external stimuli. After few days, and this segment, after few months, this segment got healed completely. So, this they have done it with all the rat animals which are treated with this kind of approach has been completely healed with this mesenchymal stem cells approach. So, first there is the creation of segmental defects. They implanted the collagen scaffold, placed this collagen scaffold. This is the scaffold and bone marrow. After 2 weeks, this mesenchymal stem cell slowly it is invading the scaffolds, and then they inject DNA and microbubbles, which has bone morphogenetic protein, and they apply ultrasound, the external stimuli. Now all the DNA will get into the cells. Now it will start bone repairing process. See how beautiful is this approach. So, where they have scaffold, cells and growth factors as well. All the three combined together and used for the repair and regeneration of that big bone defect. In the next session, we will be dealing about the growth factor based approach and the commercially available bone grafts, and what are the current trends and future direction of bone tissue engineering.